Alright, so we're starting up now with our first lecture on an overview of animal diversity, and even though the slide says Principles of Bio 2, I actually use this lecture in a lot of courses because it's a good opening chapter to start talking about how animals are classified and some of the really big developments that made them the complex group that we know them today. So even though it says, Bio 12 or 1520, Principles of Biology 2, don't worry about that. This lecture is used for a lot of classes, and so it's just kind of an opening look at the animals. So, moving into the animals as a group, this first slide is going to have several features that all animals share without exception. And so, number one, animals are always going to be heterotrophic. And so, heterotrophic, as we've talked about before, means that animals are unable to manufacture their own food, whereas plants are autotrophic and able to turn things like non-living carbon dioxide, sunlight, and water into sugar. Animals can't do that, right? So animals have to get their carbohydrates by consuming someone else, either someone who is alive, if they're a predator or a parasite, or someone who was alive, if they're a detritivore, decomposer, or something that breaks down dead organic matter, something like that. Herbivores also are heterotrophic, right? Because they're feeding on plants, and plants are, of course, alive. Animals are multicellular, so a couple parts of that definition. This definition I found often presents some issues because it's seemingly a very straightforward definition, but it turns out there's more than just one part to it. So for an organism to be multicellular, two things have to be true. Number one, obviously it has to have more than one cell, but secondly, those cells have to be divided up to form tissues. So in other words, some cells are doing one job and other cells are doing other jobs, and if you have that going on in an organism, then it is truly multicellular. And so animals are always multicellular. They are never single-celled. There are a few that are colonial, which is kind of like a gray area between single-celled and multicellular, and we'll see a few of those kind of early on when we talk about specific animal groups. But all animals, at the very least, are cooperative in their cells. Animal cells never have cell walls, right? So it'd be really hard to move. And movement is one of the hallmarks of animals that we'll talk about here shortly. But it'd be really hard to move if all of your cells were locked together with cell walls. So we know that animals never, ever, ever have cell walls surrounding their cells. They live in just about every kind of habitat you can imagine, from those that live in the air to those that live on land to those that live below ground or saltwater or freshwater, moving water, standing water, whatever. Just about any habitat you can think of, you'll find animals living there. So when we say that animals are successful, that relates not only to the number of species there are. Animals are the most diverse group of organisms out there, and that's a little bit misleading because animals have been also studied more than other groups than, say, like bacteria or fungi. But animals are a very diverse group, so they're successful from that standpoint, but they're also successful in the fact they've colonized a huge array of habitats. Now, the vast majority of animals are invertebrates. The last 1% there of organisms that are animals are things like fishes, which I know sounds kind of silly, but fishes actually is an appropriate term to use if you're talking about more than one species. So fishes, amphibians, reptiles, and mammals are the vertebrates, and birds are also classified as reptiles. They're kind of our other group there. Those are all vertebrates, but they only make up about 1% of all animals. The other 99% or so are invertebrates, so things like insects, and things like mollusks, and things like roundworms or segmented worms, and all the other kinds of animal groups that don't have a backbone are the invertebrates. Animals can always move at some point during their lives. Now, I have to kind of qualify this a little bit because animals don't always move throughout every stage of their life, but at some point during their life, all animals have at least some capability for movement, and in order to have that, they have to have nervous tissue and they have to have muscle tissue. All right, so muscle tissue to actually cause the movement, but nervous tissue to control the muscles, which then cause the movement. So minimum, some degree of neuron function, some degree of muscle function, and that's going to give you the ability to move. Now, there are some animals that don't move throughout part of their life. We call those animals sessile. So sessile is Sessile. Sessile means attached or not moving. So when you find a sponge anchored to the bottom of the ocean or a coral on a reef, those animals are non-moving, but at some point during their life they did move. For both of those two organisms, they moved when they were juveniles, and they eventually find a place to settle down and do so. But all animals do move at some point during their life. Animals are biased heavily towards sexual reproduction. Now, we do reproduce asexually, and even in humans, this is the case. When our cells replace themselves, it is through asexual reproduction, which we call mitosis. But animals are also very much oriented towards sexual reproduction, where we produce what are called haploid gametes. So haploid is a single set of chromosomes. The gametes in animals, sperm cells and egg cells, are haploid. So each sperm cell carries a haploid set, each egg cell carries a haploid set, and so when those two get together, they form a diploid cell, which has two sets of chromosomes, which makes up the rest of the life cycle. So the animal life cycle is made up of single-celled haploid gametes, and multicellular diploid parts of the life cycle dominate the vast majority of it. All right, so that's a feature which distinguishes us from things like fungi and things like plants. Fungi actually have a more heavily haploid life cycle. Plants are actually more like 50-50 haploid-diploid life cycles. So animals are very much biased toward diploid, with the haploid stage only forming the gametes. And again, those are single-celled in our life cycle. We will take a look at embryonic development in this chapter because that's a really good feature to use when it comes to classifying animals, is how they develop embryologically. So when we look at the traditional animal classification, which is what we're going to take, the approach we're going to take in this chapter, what we're going to find is that within the kingdom Animalia, there are about 35 or 36 phyla. Now, the reason why there's a range there is because not everybody classifies the animals the same way. And this is actually really typical in taxonomy or the science of classifying organisms, is that people will often disagree on which groups should be placed with which other groups, or which groups should be separated out, or how exactly things are related. So sometimes if you ask one taxonomist, they might say there's a certain number of species or families or orders, or in this case phyla. If you ask somebody else, they might give you a very different answer. So depending on who you talk to, traditionally, and there is a newer, more contemporary scheme of classification, which we're going to talk about toward the end of this chapter, but in the traditional mode of classification of the animals, there are about 35 or 36 different phyla. And we'll talk about some of those as we go on in this chapter. All right, so just some pictures, some show and tell here. Animals are heterotrophic. All right, so we've got this grizzly bear trying to catch a salmon, so feeding on another organism makes it heterotrophic. Animals are able to move with muscles and neurons. For the most part, animals are multicellular, with a couple exceptions that we'll talk about in this chapter a bit later on. They don't have cell walls, ever, ever, ever. Very diverse in form. This happens to be a millipede in this picture, which is a type of arthropod. It's in the same phylum as the insects and spiders and ticks and mites, and a few other things as well. Crustaceans are in this group as well. So very, very diverse in form. Lots of different habitats. So in this picture we have a brittle star, which is a type of echinoderm related to starfish, sort of a cousin of starfish. Different types of tissues. That goes back to being multicellular. In other chapters, in either Principles of Biology or Intro Biology, we talk about different tissue types in animals. There are 4 of them. So animals have epithelial tissue, connective tissue, muscle tissue, and neurons or nervous tissue is kind of the 4th option there. We have this characteristic development that we'll talk about a bit later in the chapter. And of course, gratuitous sex picture here, tortoises mating sexually. That is usually the case as well, although there are some of the more primitive animals which can reproduce asexually and actually produce new organisms, as opposed to replacing tissues like humans do through asexual reproduction. There are some organisms, some animals, that actually produce offspring through asexual reproduction as well. So the approach that we're taking in the rest of this chapter is going to go something like this. We are going to pretend that somebody has just brought us a brand new, never before seen animal. It's in a box, and nobody has ever seen it, and it's our job to classify it. So the kinds of things that we're going to use as a taxonomist to classify this animal are the presence of tissues, if any, what type of symmetry it has, if any, what kind of body cavity it has, if it has one, what kind of development it goes through, and then finally whether or not it's segmented. And those are kind of the really big developments that occurred in the evolution of the animals, going from what essentially were protists that kind of hung out together and cooperated a little bit, to eventually multicellular animals. And so we'll talk about what each one of these terms means, and talk about what it's important for as far as allowing the animal to perform different kinds of functions. So for the very first thing that we're going to look at, the first division of animals is whether or not tissues are present. All right, so basically what we're saying here is, within this organism, are some cells doing some jobs and other cells doing other jobs, or are they all basically doing the same thing and each one's kind of looking out for itself and they just happen to hang out together? Right? So either there's specialization or not. So if there's not specialization, we say this animal is a parazoan. All right, so I like to talk about parts of words a lot in my classes because I find it really helps people understand what they mean, and sometimes even helps you figure out new words before anybody ever defines them for you if you just know what the roots of the words mean. So this is actually a really good example of that, because whenever you see the prefix para, it means kind of, sort of, right? So if you're talking about a paramedic, right? A medic is a doctor, more or less, right? So a paramedic it's kind of sort of a doctor, but not really a doctor, right? They're kind of sort of doing some of the same things, but not completely, right? So if somebody is a paralegal, legal means lawyer, paralegal means kind of sort of lawyer, right? So they're not really fully a lawyer, but they're doing some of the same duties that a lawyer would do, so they're a paralegal. In this case, we have a parazoan. And so whenever you see the root zo, that means animal. So parazoan means kind of sort of an animal, right? So in this case, parazoans are the most primitive of the animals in that they do not really fully have tissues or organs. And the only animal group like that is the phylum Porifera. The phylum Porifera refers to the sponges. Like we see in this picture, these are yellow tube sponges. These are sponges as well, actually. They are colonial animals. Now, this is a kind of an exception that's made for the sponges because they're really not multicellular. They really don't truly have tissues, even though I just finished telling you guys that to be an animal you have to have tissues. Sponges don't really fully have tissues, but they're a little bit more than just colonial as well. They're kind of a gray area. They're more multicellular than a protist, but they're less tissue-based than an animal. They're kind of somewhere in the middle, and so traditionally taxonomists place them in with the animals as being the most primitive, most basic, simplest form of animal that there is. All of the other animals that are out there are called metazoans or eumetazoans. Sometimes you'll see that as well. And so for the metazoan animals, not only do they have tissues and specialization and those kinds of things, but they're also going to have a definitive shape. They're going to have some sort of symmetry. They're going to have some embryological development. All the other things we saw back here only happen for the animals that have tissues. For animals that don't have tissues, like sponges, they don't have symmetry, they don't have a body cavity, they don't develop the same way other animals do, and they're non-segmented. If an animal does have tissues, that opens the door for all of these other things to occur. But not having tissues means that sponges are in kind of their own category of animals. Now, another term you might sometimes come across is the term protozoan. Now, proto means kind of like before something, or an early version of something, or an early form of something. So again, zo is animals. Protozoans are like the pre-animals. They're the protists that are animal-like. They're animal-like protists. And all that really means is they're protists, so they're not multicellular, but they're single cells that still move around, kind of like an animal does. So protozoans are things like amoebas and paramecium and euglena and those kinds of things that we think of as moving, but they're single-celled and therefore they're not animals. So those are protozoans. And why are they the early animals here? Well, because those protozoans, one of those groups, is the one that eventually gave rise to the animals. So they're the pre-animal protists, essentially. So with tissues then, we have our parazoans, all right, sponges, the simplest animals, have no tissue level specialization. They don't have epithelium, they don't have muscles, they don't have nerves, they don't have connective tissue, they certainly don't have organs, because you can't have organs if you don't have tissues. So they are very, very, very simple. Now, we tend, I think, as complex animals, we're humans, complex animals, we tend to kind of bias our thinking against animals that are simpler. All right? We tend to think that we are more adapted or better suited or a better fit to the environment because we're more complex and we're fancier and those kinds of things, more specialized. That's not always the case, because sponges actually, because they're simple, are able to do things that other animals can't do. So for example, if you were to take a sponge and actually pass it through like a screen or a filter and separate all the cells from each other, those cells, if you place them back in the vicinity of each other, can actually reform the original sponge. Right? Now, that is something that only a simple animal can do. If you tried that with your cat, okay, don't do that. Don't do that with your cat. If you separate your cat at a cellular level, that's going to be a really sad day, and a bunch of people are going to be really mad at you. So don't do that. But even if you did, of course, the animal wouldn't survive, right? Because the cells are specialized. The brain cells do one job, and the liver cells do another job, and skin cells do a different job. And if you separate all those cells from each other, they are going to die, because each cell has its own job, and it relies on all the other cells around it to help it to survive. So if you separate those cells, when they lose their companions, they just all die. If you're a simple animal like a sponge and you separate those cells, because every single cell is essentially doing the same job as every other cell, they don't care. They're perfectly fine. And if they happen to come into contact again, they will eventually reform the structure that we think of as being a sponge, but they're not going to die as a result because they're simple. Another feature of being simple is that they're not especially picky on their requirements. And sponges, in fact, can live in environments that other animals can't live because sponges are so simple. So, for example, sponges can live in deep ocean environments, or what we call marine environments. When you hear me say marine, that means saltwater, ocean kinds of places, right? Deep water ocean environments are some of the most stable environments on Earth. They hardly ever change. It doesn't matter what's going on land. If an asteroid just hit the Earth and ended essentially most of the dinosaurs, the animals living in the deep ocean couldn't care less. They don't even realize anything happened because the deep ocean is so stable. It's always dark, it's always cold, there's always a lot of pressure. And so if you can survive in that environment, you can enjoy a relatively stable existence because your environment is not really going to change that much. On the other hand, if you're more specialized and you've got a very specific habitat with very specific food requirements and sorts of things like that, then it's going to be a bit more difficult if the environment starts to change around you. All right? Now, another feature of very well developed and specialized animals is that their cells differentiate themselves. And like we talked about with the cat example, some of the cells become blood cells, and some of the cells become pancreatic cells, and some cells become cells on the tongue or whatever. Once those cells differentiate, they can't go back and undifferentiate. Once they've committed to becoming a certain kind of cell, they have to stay that kind of cell. If you're a simple animal, that's not the case. Because again, every single cell essentially does the same thing as every other cell, so that if you lose a part of the organism, if part of it breaks off and floats away, the cells that are left behind are perfectly fine because they're doing the same job anyway. And again, that's a benefit. And one of those features, which is one of the reasons why sponges have been around longer than any other animal, is because they are very unspecialized and they can tolerate a wide range of conditions and habitats that are very, very stable. Another feature of sponges' success, even though they're not a very diverse group in terms of species, but they've been around for a long time, is their mode of feeding, which we'll talk about a bit later on as well. They essentially are able to take particles of food out of the water and use that for energy. And of course, if you're living in the water, there will always be particles of food in the water that you can feed on. So very, very abundant food supply as well. All right, so our next feature that we're going to look at in this section is on symmetry. And so if a shape or a structure, or in this case an organism, is symmetrical, that means you can sort of divide it into two or more equal halves. So using like an imaginary line, you can divide the organism into two equal halves. And so there's basically two options here. But the thing to remember about symmetry is that in order for an organism to be symmetrical, it must first have tissues. Sponges, because they don't have tissues, are therefore asymmetrical. They do not have any sort of symmetry at all. Every other organism, every other animal that has tissues, which is all of them, because they have tissues, they will also have symmetry. All right, but there are two different kinds of symmetry we can look at. One of them is called radial symmetry. So radial symmetry occurs essentially in organisms that are round and where you can draw an infinite number of lines of symmetry through that organism and get two equal halves. So we'll take a look at a few examples, but a jellyfish or a coral would be a really good example of a radially symmetrical animal. Essentially, radial symmetry works really well for organisms that are attached or sessile. If you don't need to move around a whole lot, if you don't need to have a well-specialized differentiated head, and being round is okay, especially if you're attached, then that's a really good place to be radially symmetrical. But it turns out there's only one phylum of animals that is radially symmetrical, which we'll talk about here a bit later on. Bilateral symmetry is when an organism only has one line of symmetry, and we call that line the sagittal plane. Right? The sagittal plane is the imaginary line that you can draw down through the middle of an organism and get two equal halves. So humans are bilaterally symmetrical. If you were to draw an imaginary line from the top of your head down, you would get a left side and a right side that are more or less symmetrical. Now, if you draw any other line of symmetry, if you draw a line of symmetry through your waist, for example, you've got a top half and a bottom half that are very different, not symmetrical, right? Any other line of symmetry except for that first one, the sagittal plane that divides you into left side and right side, any other line of symmetry will not produce two equal halves, so therefore humans are bilaterally symmetrical. And most organisms, most animals are bilaterally symmetrical. The answer to this next question is yes, they can be both radially and bilaterally symmetrical if they change throughout their lifetime. So what you'll find sometimes is organisms that start off radially symmetrical and eventually transition to bilateral, or start off bilateral and transition to radial. It can go either way. As long as they are bilateral for even 5 minutes in their life, you classify them as bilateral. They could be radially symmetrical for 99% of their lives. If they're bilateral for even a minute, then you classify them as bilateral. Okay, so it's a very specific criteria here. If bilateral ever, classify them as bilateral. All right, now once we go beyond that, once we go and look at the bilateral animals, we can then start classifying them based on whether or not they have a coelom. Right, so this term down here is pronounced coelom, and this is a true body cavity. There's a couple of other options we could talk about there as well, but coelomates are the most abundant groups of animals. We, humans, vertebrates, are coelomates, which we'll talk about more what that term means in a later section. All right, so radial symmetry, again, just to kind of recap, all the body parts are arranged around a central axis. Because they're essentially circular, there are an infinite number of lines of symmetry you can draw through the organism to get to equal halves. For bilateral symmetry, you have a right side and a left side, and that also, having a right side and left side also confers a bit more directionality to the body as well, as we'll see on this picture with this turtle. This sea turtle has a sagittal plane dividing it into a left side and a right side. Whenever we talk about right side and left side anatomically, we're talking about it from the organism's perspective. So from this turtle's perspective, this is its left side, and over here is its right side. So instead of saying this is right and this is left, because that's our perspective as we look at the animal, we always go by the organism's perspective, the patient's perspective, so to speak. Anatomical right and left is what we're referring to. So because there's a right side and left side, that means there's also an anterior, which is a front, and a posterior, which is a back. We have a dorsal, which is the back surface, and we have a ventral side, which is the belly surface. And so the only way we can have that directionality is if the animal is bilaterally symmetrical. Now for a sponge, sponges, like I mentioned, are asymmetrical, so there is no front, back, top, bottom, right, left. They're just essentially a blob. Now some sponges can look like they're somewhat symmetrical, but they're not really. Cnidarians, which is the next group that we'll talk about, are radially symmetrical. So cnidarians are things like jellyfish and hydras and corals and anemones. Box jellies are another one that probably a little bit less familiar with, but those are all radially symmetrical animals that have all these lines of symmetry you can draw through them, like here or here or here. But again, because they don't have that one line of symmetry, they don't have a definitive front, back, top, bottom sort of side to them. Now in this picture we have a few examples to think about. Now these are anemones up here. And anemones are cnidarians that are radially symmetrical throughout their entire life. These guys though, these spiky guys, are called sea urchins, and they are echinoderms. They are distant cousins to things like starfish and so forth. These guys in this picture, they are radially symmetrical because you could draw lines of symmetry here or here or here or there. Essentially an infinite number of lines of symmetry. So you might think they're radially symmetrical, but if you look at them in their juvenile stage, they actually are bilateral. All right? So again, like I said before, if they're bilateral ever for even a moment in their life cycle, we classify them as being bilaterally symmetrical. All right, so the other advantages of being bilaterally symmetrical, aside from having directionality, is it makes you a lot more specialized in your body as far as your nervous system and your musculature. You can have a bit more of a centralized nervous system if you're bilaterally symmetrical. So centralized nervous system means you're going to have things like a brain and a spinal cord. Eventually, as we move on up through the animal groups, the more advanced animals have more centralization because they are bilaterally symmetrical. They have what's called cephalization. The term cephalization refers to the fact that they have a definitive front end, a head, essentially. The front end of an animal is important for a couple of reasons. It's typically where you find a lot of the really important sensory structures, so eyes and ears and nose in some animals, for example, are in the front end, the head. But the downside to that is if the front end is damaged, because there is a lot of centralized nervous system there, a lot of sensory structures there, food intake also occurs at the head as well. If the head is damaged, then that very frequently will be the end of the animal. So it's a bit of a double-edged sword where having a cephalized animal provides some benefits. It could also be a disadvantage if the front end is damaged. All right, so again, animals that are bilaterally symmetrical, that are cephalized, oftentimes are more mobile. They can move in an oriented way toward a stimulus or away from a stimulus because they have those sensory structures in the front end. All right, so we're going to cut off this section here, and we will start up in just a second with our second section on animal diversity.